Have you ever wondered how microbiologists identify different types of bacteria under a microscope? One of the most widely used laboratory techniques for this purpose is gram staining. It is a simple, rapid, and highly effective differential staining method that helps distinguish bacteria based on the structure of their cell walls. In this video, we will learn the principle, procedure, and interpretation of gram staining, step by step. What is gram staining? Gram staining was developed by the Danish bacteriologist Hans Christian Gram in 1884. It divides bacteria into two major groups, gram-positive bacteria, which appear purple, and gram-negative bacteria, which appear pink after staining. This difference is due to variations in the structure of their cell walls. Principle of gram staining. The principle of gram staining is based on differences in bacterial cell wall composition. Gram-positive bacteria possess a thick peptidoglycan layer. During the staining process, this thick layer traps the crystal violet iodine complex, allowing the cells to retain the purple color. In contrast, gram-negative bacteria have a thin peptidoglycan layer and an outer membrane. During decolorization, the crystal violet iodine complex is removed, making these cells colorless until they are stained with the counterstain, safranine. As a result, gram-positive bacteria appear purple, while gram-negative bacteria appear pink. Materials required. The materials required for gram staining include a clean microscope slide, a bacterial culture, a sterile inoculating loop, a Bunsen burner, crystal violet, grams iodine, alcohol or acetone, safranine, distilled water, bibulous paper, and a compound light microscope with an oil immersion objective. Step 1. Smear preparation. Begin by placing a small drop of sterile water onto a clean microscope slide. Using a sterile inoculating loop, transfer a small amount of bacterial culture to the slide. Spread the culture gently to form a thin, even smear. Allow the smear to air dry completely before proceeding. Step 2. Heat fixation. Once the smear is completely dry, gently pass the slide through the flame of a Bunsen burner 2 or 3 times. Heat fixation kills the bacteria and firmly attaches them to the glass slide, preventing them from being washed away during staining. Avoid overheating, as excessive heat may distort the bacterial cells. Step 3. Primary staining with crystal violet. Flood the smear completely with crystal violet. Allow the stain to remain on the slide for approximately 1 minute. Crystal violet penetrates all bacterial cells, staining them purple. After 1 minute, gently rinse the slide with distilled water to remove excess stain. 4. Treatment with grams iodine. Next, cover the smear with grams iodine for 1 minute. Grams iodine acts as a mordant. A mordant is a chemical that strengthens the binding of a stain to a cell or tissue, combining with crystal violet to form a large crystal violet iodine complex inside the bacterial cells. After 1 minute, rinse the slide gently with distilled water. Decolorization. This is the most critical step of the gram staining procedure. Decolorization is the process of removing the primary stain, crystal violet, from certain bacterial cells using a decolorizing agent, usually alcohol or acetone. Apply alcohol or acetone for approximately 10 to 15 seconds. In gram-positive bacteria, the thick peptidoglycan layer retains the crystal violet iodine complex. In gram-negative bacteria, the alcohol dissolves the outer membrane, allowing the crystal violet iodine complex to escape. Immediately rinse the slide with water to stop the decolorization process. Overdecolorization or underdecolorization may produce incorrect results. Step 2. Counterstaining with safranine. Now flood the smear with safranine for 1 minute. Safranine is used as a counterstain after decolorization. It is essential for gram-negative bacteria because they lose the crystal violet stain during decolorization and become colorless. Safranine stains these bacteria pink, making them visible under the microscope. Gram-positive bacteria, however, retain the crystal violet stain because of their thick peptidoglycan layer. Although safranine is applied to them as well, they remain purple because the dark crystal violet masks the pink color of safranine. Thus, safranine helps distinguish gram-negative bacteria from gram-positive bacteria. After 1 minute, rinse the slide gently with water. Blot the slide dry using bibulous paper. The slide is now ready for microscopic examination. Microscopic observation. Place a drop of immersion oil on the stained smear. Observe the slide under the 100 times oil immersion objective, providing a total magnification of 1000 times. Gram-positive bacteria appear purple or violet, whereas gram-negative bacteria appear pink or red. The staining pattern helps microbiologists identify bacteria and guides further laboratory testing. Applications. Gram staining is one of the most important techniques used in microbiology laboratories. It helps in the rapid identification of bacteria. It assists physicians in selecting appropriate antibiotic therapy. It is widely used in hospitals, diagnostic laboratories, research institutions, and educational laboratories around the world.